Hello everyone. In this video lecture, we are going to discuss about miRNA. miRNAs are also called as microRNAs. These microRNAs are non-coding RNAs, which means they do not code for a protein. Okay, so these microRNAs are made up of 18 to 25 nucleotide in length. These microRNAs are able to modulate gene expression, okay, by inducing degradation or blocking the translation of a target mRNA. So mRNA undergo translation in order to code for a protein. Okay, so this Hi everyone. In this video lecture, we are going to discuss about microRNA. MicroRNAs are also called as miRNAs. They are non-coding, which means they do not code for any protein. These microRNAs are made up of 18 to 25 nucleotides in length. The microRNAs are able to modulate gene expression by inducing the degradation or blocking translation of the target mRNA. As you know, DNA is made up of coding and non-coding sequences. During transcription, DNA, the introns and exons in DNA, they undergo transcription to become mRNA. So these mRNA undergo splicing in the post-transcriptional modification. Once that happens, the mRNA move to the cytoplasm to translate to become a protein. Okay, so this process is blocked by the degradation or blocking by the microRNAs. So these microRNAs found in plants, animals, and viruses. This first microRNA was discovered in the year 1993 by Ambrose et al. They have identified the microRNA in the nematode called C. elegans. And the first microRNA is identified in the gene LIN4. Okay, so mutation in gene LIN4 gene can cause failure in temporal development in nematodes and structural defect, and these defects can make nematode to unable to lay eggs. Okay, so this LIN4 gene is essential for the normal development of a C. elegans. The microRNAs are highly conserved, okay, can be expressed in a tissue-specific manner. Okay, sometimes a microRNA can be expressed in one particular type of cells where they do not express in other parts. Okay, and these microRNAs perform essential functions in regulating diverse cellular processes, including development, differentiation, and disease progression in animals. So far, more than 1,000 human microRNAs have been identified, and these microRNAs have been sequenced. And it is estimated that 1/3 of genes are regulated by microRNAs. And one microRNA can regulate the expression of many genes. Okay, so microRNAs are transcribed by RNA polymerase II. In prokaryotes, there is only one RNA polymerase is present, whereas in eukaryotes, you have 3 RNA polymerases: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Here, the microRNA is transcribed by an enzyme called RNA polymerase 2, okay, with the help of transcription factor 2D. This transcription factor 2D binds to the Tata sequence of a DNA. And this enzyme, RNA polymerase 2, is located in the nucleoplasm of the cell. And it is a complex of 550 kilodalton enzyme, and it has 12 subunits. And coming to the structure of microRNAs, there are two structures in microRNAs: primary structure and secondary structure. Okay, the primary structures are a double-stranded RNA molecule, and it contains 70 nucleotides sequence. And this microRNA in a primary structure is having a hairpin loop. So this hairpin loop is flanked by a region which is required for the efficient processing of the microRNA. As you can see, this is the primary structure of a microRNA. And the microRNA undergo processing in two different compartments. First one in the nuclear compartment, and after that it undergo processing in the cytoplasm. Okay, let's see one by one. In the nuclear processing, the primary microRNA transcript is cleaved by endonuclease 3 enzyme, which is called DROSA, and this enzyme is accompanied by another protein called a PASA. Okay, this DROSA and PASA, they form a complex, and this complex is named microprocessor complex. This complex is playing a vital role in processing the primary RNA, microRNA, to become a secondary microRNA. So in this complex, PASA orients the catalytic RNase 3 domain of the DROSA enzyme to liberate hairpin loop from primary microRNA. And by that, it cleaves the RNA about 11 nucleotides from the hairpin base, and which results in the product of two nucleotide overhangs at the 3' end and the 5' end. At the 3' end, you have a hydroxyl group, and in the 5' end, you have a phosphate group. And this often termed as a primary micro or premature microRNA. Okay, as you know, once the DNA get transcribed, you have a premature mRNA. So likewise, in microRNA also, you have premature microRNA. So this premature microRNA undergo processing in the nucleus. Okay, and so once this pre-mRNA is formed, this pre-mRNA hairpin are exported out of the nucleus in the process involving a protein called exportin 5. Okay, and also the cytoplasmic transport, which is from nucleus to cytoplasmic transport, it requires energy dependent. For this, it uses DGP bound RAN protein. As you can see, this DROSA and FASA, they cleave the primary microRNA to become a premature microRNA. So these two regions gets cleaved and you retain this region, and this region is exported out of the nucleus with the help of exportin 5 and RAN protein, okay, which is mediated by GTP. And once this happens in the cytoplasm, again it undergo processing. The microRNA undergo processing. In the cytoplasm, the pre-mRNA microRNA hairpin is cleaved by RNase 3 enzyme, which is called dicer. Okay, it is also called as endoribonuclease. Okay, this dicer, it interacts with the 3' end of the hairpin and cut away the loop joining the 3' and 5'. And then these arms provide imperfect microRNA. Okay. And you have two strands. Okay, either one of the strand duplex act as a functional microRNA, and that microRNA is incorporated into the RNA-induced silencing complex. Okay, this RISC complex where microRNA and its mRNA target, they interact. So this is the mature microRNA. After either one of this strand joins with the microRNA, and then they become a mature microRNA. So this mature microRNA bind to the target mRNA to interact for the mRNA target cleavage, translational repression, and mRNA deadenylation. Okay, the other strand in the microRNA, it undergo degradation. The selected strand is incorporated into the risk complex on the basis of its thermodynamic stability and weaker base pairing, which is related to the other strand. Okay, so the risk is also known as a microRNA ribonucleoprotein complex or miRNP complex. The mature microRNA is a part of an active risk containing dicer and many other associated proteins. Okay, so the member of agronate protein family is a central to risk function. Okay, without this, the risk cannot function efficiently to carry out these processing in the cytoplasm. So these protein complexes, they bind to the mature microRNA and then they interact with the target RNA. So this is how a microRNA is get processed nucleus as well as in the cytoplasm as like mRNA. So as we know, mRNA, when they get when the DNA gets transcribed, they produce mRNA. So that mRNA undergo post-transcriptional modification in order for the mRNA to export to the cytoplasm. Once they are in the cytoplasm, the mature RNA undergo translation and that produces protein molecules. The same mechanism, the microRNA processing takes place in the higher eukaryotic organisms. Thank you for your patience.